I don't mind going first if you want me to. So hi, everyone. My name is Shelby. I am a master's student in the neuroscience program at the University of Guelph in Ontario, Canada. And my research focuses on the effects of positive early life experience in protecting against Alzheimer's disease and brain damage, and the memory and cognitive impairments that we see in Alzheimer's disease specifically. So I'm very excited to be here today, hopefully answer some interesting questions that you may have about research or grad school in general. I am just about to finish my first year of my master's, so I'm still new to the field, so I would be happy to provide any information that you guys would like.
Hello, everyone. I'm Carol Gilbert, and I am a little bit further on in the career. I finished my PhD 3 years ago, so I am still an early career researcher. I am originally from Brazil, and I finished there my master's, my PhD, and then I came to Australia, where I am now, working as a postdoc for 3 years, as I mentioned, and I work here with the microbiota, gut-brain axis, so with the microbes that live in the gut, and how targeting these microbes can be useful for a few diseases, mainly Huntington's disease and schizophrenia. Those are the main diseases I'm studying at the moment. Again, also happy to answer any questions you all might
have. I'm excited to be following up those two interests because they're so interesting. I already have questions for both of you. But yeah, so anyway, I'm super excited to be here too. My name's Jackie. I am currently a fourth-year PhD student at Brown University, and I am basically I'm doing my PhD in pathobiology. But I am both in like the neuroscience department and the EEB department and a little bit of this aging department. So it's a little bit spread out. But currently I study basically the innate immune system and its role in neurodegenerative disease as well as behavior. And I'm just really, really interested in trying to find out like how we could maybe target some of these immune factors to get some sort of targeted therapy. That's like sort of my main goal. I also sort of have a background in it's a weird path, but I started out as an environmental science major at Berkeley, where I did my research on like ecology and evolutions of lizards. So it's very looking at like niche habitats and how they're used by different species. So that really gave me like a framework for how I view all of my research through an ecological lens where nothing is separate and kind of compartmentalized. I like to think about it as like everything's sort of connected. And, you know, the gut microbiome is a great example of that. And then after that, I went and did I took 4 years off and I started to do clinical immunology as like a research assistant at UCSF, where I studied basically just the role of Tregs in a lot of diseases, like type 1 diabetes was my main project. And then I moved on to a couple other different things, drug development with Pfizer to try to target rheumatoid arthritis. And also to try to like develop some other immunotherapies for rheumatoid arthritis. And then the last kind of thing that I'm doing now, I guess, is trying to combine all of those things into my own research at this point. So sorry, my intro is a little longer. I just wanted to give you guys a background of what maybe I could answer. And I'm super happy to answer any questions about either my science or perspectives, which I think is really cool to talk about and big picture ideas. And then also just any questions you have about like grad school or taking time off or like what you might want to do, because peer mentorship and mentorship in general is just something I have a big passion for. So I'd love to answer any questions about that. Thank you.
Sorry, if I can complement also, I loved your intro, Jackie, pretty complete. If I want just to complement to everyone knows also other things that you can also ask me, because I also changed a little bit of my field from my PhD, master's, and graduation. studies to when I came to Australia. So I used to work with bipolar disorder and one specific Purinergic receptor to target and see if we can find a new treatment for bipolar disorder. Always I work with animal models, but also in this, in my PhD, I also work a little bit with patients. So yeah, happy to answer also how, you know, you have to sometimes change your field, as Jackie mentioned, and how challenging this is, but also how exciting can be, and yeah, can complement all your skills.
Wonderful introductions, and we're very happy to have you here. But Sydney Lay is our SN admin, and she will be hosting our Q&A. So Sydney, take it away.
All right, I know some of you already went over this, but could you talk more about how you got into your current field?
I can start. So I did a Bachelor of Arts in Science at Guelph, and I originally just wanted to stick more to the psychology and more of the behavioral side until I took my first neuroscience class, and I was introduced to this whole new field that I thought was super, super interesting, because I was interested in the behavior side, but also the more applicable structural side. And so I did some research in a rat and a mouse lab looking at memory reconsolidation. And I just thought it was super, super interesting. And I wanted to kind of introduce myself to that field a little bit more and see if I can maybe make a career out of it. And so I decided to stay for grad school in the exact same lab that I did my undergrad thesis in. And that's where I am now. And I am loving it. And I am planning on staying also for my PhD, hoping to maybe stay in the Alzheimer's side, but also maybe go back a little bit to the rats and the mice, looking at memory reconsolidation and memory updating. So that's kind of where I'm at. I'm sort of still in the middle trying to figure it out, but I am enjoying the journey so far.
So kind of funny story, like being in the right place in the right time. So as I mentioned, I was working with my PhD with bipolar disorder. I was invited to go to a conference in the US to present a poster. I was presenting my poster, but it was a kind of a funny conference full of medical doctors. So not exactly focus on basic science. So my poster was the only one about basic science, and my current boss was the only speaker that was talking about basic science. So pretty much we matched. He came to my poster, we chat for 2 hours. I was in the middle of my PhD. He invited me to, he asked me my plans for the future. Of course, I took the opportunity. I said I was seeking some postdoc abroad. And then he said, well, perhaps, you know, you can join my lab. Let's keep in touch. And then I was looking for postdoctoral fellowships. And then one of a very important, very good fellowship, one of the criteria was that I should apply with a project that wasn't related with anything that I did for my PhD or anything that my planned boss was working with. So it was a huge challenge. And then we talked, and of course, the gut microbiota is a very, very interesting topic and is a current, and 3 years ago was definitely a hot topic. So we agreed that that was something that we both shared interest. And then I didn't get the fellowship, but we kept the idea because we really fell in love with the project. And then, yeah, I got another fellowship, but yeah, we kept this idea. So sometimes, you know, life puts you in your right direction. And now I love it. I'm definitely passionate about it. I don't plan to change, at least in this near future. So.
I mean, I would like to echo that through my story, I guess. So I mean, I don't know how many of you were on beforehand, but me and Shelby were talking a little bit about, you know, just how we ended up here and sort of our backgrounds and stuff. But basically, yeah, I started off as someone who was just in environmental science at UC Berkeley because I really had a passion for well, in high school, I actually had this weird internship with the San Francisco Public Utilities Commission where they were doing a lot of work on basically how to build dams that were safe for fish and salmon to be able to swim back to. And so that was sort of my first taste of biology that I really liked. But I also liked the social aspect or the but basically like the policy aspect of it as well. And so that's what I initially thought I would be doing. Then when I got to school, I was studying like environmental science, taking the courses, but I needed to get a job because I needed to have some extra money to be able to live. And I got really lucky. So I was just searching for jobs, and I didn't even know labs existed on campus at that time. Like I thought I didn't know that was an option. And I saw on like the student webpage or the student works site webpage that there was this job opening as like a lab assistant in an immunology lab. And it was really basic work where it was like basically I was taking out trash, cleaning off people's like desks, like restocking everything. But it was paying super well. And that was like very, very, very like, I can't even believe that happened, because without that, nothing else would have occurred for me, at least not in the same way. And so I worked there for like 3 years, so for my second to fourth year, and still while doing environmental science stuff. But I was slowly picking up on just like, I would just go to the lab meetings. I would try to go to like all of the extra stuff and ask people about their research as I was like, restocking their area. And it was just really fascinating to me. And so I did my thesis. I was invited to do my thesis in that lab, but I couldn't because I was already doing my thesis in a different lab because of my environmental science degree required a thesis. So I basically just found a professor on like the university website of professors in EEB, and she was new. Her name was Dr. Erica Bree Rosenblum, and she had just transferred from Idaho. And I just sent her a cold email being like, hey, like I really need to do a thesis project. Your work looks really cool. Could we chat about it and maybe my potential doing something in the lab? And because she was new and she was looking for someone, she just sort of was like, yeah, like you could go to White Sands with these grad students of mine that are going to be coming back from Idaho. So I got that ecology research, and I did that for another summer. So I did that my summer of my junior year, the summer of my after my senior year, after graduating. But then I was looking for a job, and there's just not a lot of jobs that are paying super high in ecology and evolution right now. There's a lot of seasonal work, but also I applied to grad school, and I basically got into one, but kind of in a weird way, and it was on fish ecology, and I was just like, I don't know if this is like what I really want to do. So I applied to a ton of jobs. Like I just threw out my applications everywhere. One in particular was at UCSF, which is what I ended up getting, which was for clinical immunology. I'm like floored that I got that job considering my background. But I basically like read all the papers that lab had been producing, and I'd always been really interested, again, because of like the past of working in that immunology lab, just in immunology in general. And so I got that job also because the professor knew the professor that I worked for before. And for that 4 years, I just like learned everything I could about immunology and clinical immunology. I worked with mice, I worked with humans, I worked with all sorts of stuff. And I feel so grateful for that experience. And then, and got a little bit of a taste of industry with that collaboration with Pfizer in drug development. But then sort of after that, I was like, okay, do I want to continue to do technical work? I had gotten a job at Genentech, like a job offer, but I was like, I don't know if I want to just do this like technical work for the rest of my life. Like I want to have my own ideas, and I want to be able to think about them and implement them and see if I can actually drive my own project. And part of that was actually coming back to sort of the microbiome and my ecology background. I was trying to stimulate some osteoclasts, which are cells that break down bone, to see if I could make a CAR. So this is like a CAR T cell, which is basically an immunotherapy for a targeted immunotherapy. You basically make a T cell that can bind specifically to whatever you've stimulated it with. And I was trying to do that for rheumatoid arthritis, and I could not get the osteoclasts to make the T cells that I was trying to stimulate them. So one of the things that I read in a paper was that P. gingivitis, so regular gingivitis, can stimulate can basically stimulate the immune system. And I thought that was super cool. And I then was like, this is ecology. Like, if I look at it, this is just ecology. And so then I was like, I need to go to grad school. Like, I need to study this. If there's no room for that here, but there's room for it there. So I looked up PIs. I mean, honestly, how it happened for me was I read a bunch of papers, and I noticed that a couple of the papers that I loved the most were all coming from Brown. And so I just reached out to the professors there. Ironically, one of the people there was a person who had worked as a PhD student in the lab that I worked in as an undergrad. And so that was helpful for me too. It's a small world. And yeah, that's just kind of how I ended up where I am. So it was like a very long story, but it's just a very weird path, I think. And just to say, like, yeah, you sometimes win some, you lose some, and you kind of, I think, can turn that into a good thing if you kind of synthesize it all together. But yeah.
Thank you. That was really interesting. Also, I just want to let everyone know that if you want to send a question and you don't want to send it in the chat with everyone in it, you can direct message me. I really love how unique all of your stories are, and I'm curious how the pandemic has affected you as it relates to your studies, research, and or career.
So luckily for me, when we work with mice, it hasn't been as much of a halt as it has been for a lot of the people that work with humans. They have to move a lot of the things online. They're not really seeing the same results that they're seeing in person because people are distracted and bored, and there's more confounding variables. So luckily, in terms of lab work, it hasn't really affected it too much. We have a smaller group of mice, so getting the mice and actually running the studies has been slowed down, but we're still able to actually complete our work. But something that has kind of sucked a bit with the pandemic is not being able to go to conferences, especially if it's in your first or second year of your master's like me, and you're excited to go network with all these people and learn about what research is happening. So unfortunately, we haven't really been able to do that, but luckily we have been able to attend some pretty cool conferences online for less of a price that we would if we had to go in person. So I guess that's a silver lining, but yeah, in terms of working with animals, it's been pretty steady. We haven't been held back too much.
Well, for me, being in Australia, as I don't know if you are update in the news, but Australia handled pretty well the COVID situation. So we currently don't have any COVID transmission amongst the population. So our life now is pretty much like as normal life. But yeah, we had a very long isolation. But me as a postdoc, I still could go to the lab and handle all the mice. So at some time, and I didn't want to, I wanted to, because I have a few students, I have 7 students amongst PhD, masters, and honors students. So at some point we had like 200 mice divided by all of them, and they couldn't go to the uni because of COVID, but I still could. So I had to do all the experiments and tests for them, just, to not, of course, kill the mice without a purpose, and also to not stop or disrupt their thesis. And a few of them were finishing their masters last year. So yeah, I had at some point a lot of work, but good thing is we didn't stop any of these experiments or this project. So now it's pretty much everything back to normal. As Shelby was mentioning, one problem is definitely that we can't go for conferences, and also, well, I studied the gut microbiome and gut-brain axis, which Professor John Cryan is a very important name in the field. And he was already accepted me to be a few months in his lab, and also I got even an hour to go and stay in his lab. I had a course that I would stay in an island in Venice. So imagine how excited I was to do that with him as well. So yeah, a few opportunities that would be amazing for my career, for my networking, couldn't happen. But of course, I am absolutely lucky for being in Australia at this time, at this moment, and during this pandemic. So yeah, I can't complain at all. I'm very, very lucky.
So I guess, yeah, I mean, I have like a two-pronged answer. One is I am very lucky to also be in like a lab that has like a lot of space. So I'm in Providence, but some labs have tons and tons of people in it. And so we would have to rotate on bases, and like I know people who would take night shifts and like work at like 3 to 5. And it was just kind of a mess in that regard. And there weren't a lot of like, it's just hard when you're all scrambling to deal with a crisis that's happening so like acutely. Like I remember the day it happened, and I was talking to my advisor, and I was like, no, this is going to be like a way bigger deal, because I had been talking to infectious disease professors about it. And my my PI is an aging person. So he was like, it's not going to affect the grad students. And I took all my flies home. So I study Tauopathes in Drosophila as a model organism. But I took all my flies home because I was like, no, I don't trust it. Like I'm doing it. And I'm super happy I did, because our incubator like broke down and a bunch of our stocks died. And that was really sad, because it takes a really long time to like build some of these genetic models. Luckily for us, I think most of ours were saved. But it was very difficult for me. Like the second prong of it is, I think also like the pandemic, coupled with a lot of the social justice issues that were happening in America at that time, made me have to rethink a lot of what my priorities were in research and why I was doing what I'm doing. Like, why am I researching what I'm researching, and who is this benefiting? So I did take some time to, and I kind of stretched it a bit to really learn more about like history of the US and history of, because I have a bunch of people who are disenfranchised that are my colleagues, and I just really wanted to know like more about it and how I could help in that. So for me, it shifted my priority from lab work to understanding these different I guess movements that are happening, so that I could be informed enough, because at the end of the day, my whole goal is to reduce human suffering if possible. And so like that's kind of just where I was at, and I'm lucky to be a grad student who had enough time and space to take that time. So yeah, it just was something for me that was like really important to learn about. And now I'm getting back onto my track and everything. So it definitely delayed my work, but I think it also made for like me to be a richer scientist. Hopefully, we'll see.
Thank you for sharing. Arushi in the chat asks how a graduate student can select a particular niche or know what interests them most.
Do you want me to go first? I can tell you like how it works for me. I'll be short. Basically, what can you not stop reading about? Like, what is it that just pulls you into it? Like, and that's honestly, like for me, it was ecology gave me the framework, immunology, and seeing the connections between like basically, the gut microbiome, which is like a little ecology in yourself. That I just couldn't stop reading it no matter what. And then my grandmother actually fell ill with Parkinson's disease, and that I was reading a lot about the gut microbiome involved with that. And I think because of that emotional aspect, and because of just like curiosity and the immune system being so complex, that's kind of just how I got to where I am, just by following my my brain's wants to read, I guess.
Yeah, I agree with Jackie. I think she used very important words, which is what a scientist actually is driven by curiosity and follow your gut instincts, I would say, because science can be very frustrating. We work sometimes 70 hours a week, sometimes most of the time. So if you're not passionate about the topic you are studying, that can be very frustrating, as I mentioned. So you have to be passionate about the topic and super curious. And because sometimes, you know, a part of working many, many hours, sometimes you work a lot and then your experiment doesn't work, or you have negative results, or all of this is very interesting if you are interested in the topic you are studying. So yeah, passion and being absolutely curious about what you're studying. I would say that's the main tip.
Yeah, I also think don't be afraid to talk to profs and see what they're interested in, what they're working on. If anything they're working on, maybe you can volunteer in their lab. Maybe you can do a course credit for some research in their lab and just kind of do an informational interview to see what work they're actually doing and if that could possibly interest you, and maybe get your foot in the door a little bit early, and then see if you're lucky, maybe later on, stay and work in their lab to pursue graduate studies as well.
Thank you. Helen asks if it is possible to treat neurodegenerative diseases with stem cells. Very hard question. MSA, Parkinson's, Alzheimer's, and if you have any information on this topic.
Yeah, so that's not particularly my area of study. I think in terms of Alzheimer's disease, firstly, it's very complex already in terms of what is causing the disease. Is it the plaques? Is it the tangles? Which cells in the brain are more vulnerable? There is a field that uses stem cells, and in rodent research, it has been proven to be pretty successful. But in order to apply that to the more broader question and to other complex disorders as well, we kind of need to do more research at the basic level to understand the causes, the different cascades that are involved, because it's a very messy disorder. Just in plain terms, it's kind of a big web of a whole bunch of things. So I think just kind of taking a few steps back and seeing what are the key players. But stem cell research, yes, it is possible, but I don't know too much particularly on that at this time.
I could take us, unless you would like to go, Carolina. Oh, okay. For me, so that's not super my specialty, but it is something that I've sort of been parallel to in some of the aging labs that are interested in how stem cells, or just cells, how they persist or how they divide, or how you could get them to slow down their cell death, things like that. So what's interesting about neurons is that they don't have cellular turnover as high as any other cell in the body. It's kind of like a kidney cell, which kind of they've been referred to as like the trash can cells, the kidney cells, because they just sort through the blood. So the stem cell thing is really fascinating, and I think it's interesting to use as a model to understand what molecules are important when we're thinking about regeneration. But I do think, and I have read a few things about Parkinson's specifically where they might insert some stem cells, but the problem is the brain is so there. We're finding out about new circuits, and we're finding about new kinds of glial cells, new kinds of neurons, and all of these, like the risk you take by putting a stem cell in without knowing exactly what I guess sort of transcription factors or molecules are necessary for it to turn into the neuron that would replace a neuron that died previously in a circuit. Like you're not sure like whether or not that will actually disrupt like everything that's in the brain. So one very big fear is cancer. So if you have a stem cell that you put in and it's just regenerating over and over, it could become cancerous. And so then you have to weigh the cost and the benefit of which would you would rather like. And I actually think that dichotomy between inflammation and cancer, or cell death and cell growth, is actually fascinating in the basic science research, but I don't know exactly how much that's been done in the clinic with success.
All right, here's a question for Ms. Howells. Yeah, do you know of some specific careers that combine environmental sustainability in psychology?
I guess, yeah. So environmental, so what was it? Environmental what? Sorry?
Sustainability.
sustainability and psychology. Was that the question? I don't know if I'm the best person to answer this, but I will say I think that there is research being done on specifically social justice in terms of social inequity in how environmental like factors, so like, you know, how we basically create a bunch of greenhouse gases, like how much that gets put off onto other nations, particularly poorer nations, people who are more disenfranchised, and that, you know, the US, for example, creates most of, I think the military creates a ton of carbon gases essentially. And so I think what is interesting about psychologically that I have seen, and I don't know how far the field has gone in this, but at least in terms of disease work, like pathobiology, we know that these sorts of toxins do actually like cause a lot more disease and can cause more neurodegeneration. And this is a big deal, especially as the lifespans in all of these different areas of the world are increasing by a lot, and the populations are increasing. So that's why you're seeing that by 2050, I think it's estimated that 60% of people over 65 will have Alzheimer's or some sort of neurodegenerative disease because of that lifespan increase. So I think what's interesting is we're having those mechanisms down. I bet you anything, I'd be interested to hear what Shelby has to say about this, is when that psychology part comes in, right? Because it's really like a feedback loop between mechanism and psychology. And I would not be surprised if we found that if there are research labs that actually study that impact of that environmental injustice on psychologies of people in nations where it's a bit poorer, where we dump all of our trash, you know, stuff like that.
Next up, we have a question for Dr. Gruber, I believe, and it asks, how come Huntington's and schizophrenias appear to have more direct correlation with the gut microbiome, and is there some interaction between the genetic component and the microbiome?
Yeah, thanks. That's a great question. So for psychiatric disorders, we have more studies showing patients with that the patients with schizophrenia, they have a different profile in the gut microbiota. And also when other groups, they perform an experiment, which is fecal matter transplant from patients and transfer into normal mice, a few of the schizophrenic phenotype was also transferred. So these kind of experiments show that, wait a minute, sometimes the profile of the gut microbiota can be related with this disease. So that's why we started thinking about that the population of the microbes that we have in the gut can be related to all the behavior and the phenotype that we can see in those diseases. So for example, I work with 2 animal models, one of schizophrenia, one of Huntington's disease. For Huntington's disease, we were the first one in the world to show in mice that the gut microbiota was different between the transgenic mice compared to the wild type mice. So because of this study, collaborators of ours, they also showed later on in patients. So we are the first group that are studying this in Huntington's disease. And this is very interesting because, as you probably know, Huntington's disease, we don't have cure yet, or not even treatment that can modulate the disorder. And it's a very, very devastating disorder. So of course, we don't think that targeting the gut microbiota will be able to cure Huntington's disease because it's a genetic disorder, right? But Huntington's disease is has a very complex symptomatology, including psychiatric symptoms and also gut dysfunction. So our hope is potentially, by targeting the gut microbiota, being able to modulate these other symptoms. So currently in my lab, we are performing fecal matter transplant from wild type mice into HD mice, seeing if you can perhaps modulate the disease by at least, delaying the onset or decreasing the impairment of cognition that this mice presents, We are also using prebiotics, which are good food for good bacteria. We are using probiotics, which are specific strains that we know are beneficial for the community, to see all of these, if in the end of the day we can modulate the behavior in these mice. Schizophrenia, we also have a transgenic model for schizophrenia, and we are also using a few approaches that target the gut microbiota to see if you can modulate the behavior. And then, as I mentioned, we work with basic science. So our hope is that all of our experiments can, suggest some clinical trials in the future, even because we are using all interventions that are already used, FDA approved, so that will be quicker translated to clinical. So,
thank you. Here's a more general question. Let me find it. Okay, what options does a neuroscience enthusiast have in terms of making a living out of the research field they choose?
I think like there's a couple options, right? Like the main ones that come from grad school at least are typically you can go into the postdoc professor, like tenured professor role if you want. And that is a more challenging road, I think, for a lot of people. And that is but I think the field of neuroscience is exploding in such a way that like it is a good field to be in if you are going to try to go that route. I think there's also industry, which is pretty big, but you would need like training to do those things. And then the other part are like startups. There are like a lot of startups for some things that you can work in, and that's like very loose. Like you can do whatever you really like. The world's your oyster if you have enough funding. And then I don't know for neuro, like there's a lot of psychom. I mean, look at what Simply Neuroscience is doing. Like these kinds of things can be built up, and I think maybe should. But I don't really know much more about like. I mean, I actually currently am thinking about like what do I want to do with myself in the future, and I don't really know, but it is a skill set where you can learn so many different. Neuroscience covers so many different kinds of skills. Like people learn software engineering, they learn like basically how to like understand psychology to some degree. They learn behavioral techniques and molecular techniques. It's just like an imaging techniques, and all of these things are transferable skills that you can really use in other situations. But these are all, I guess I'm speaking more to like if you're going to stay in science versus like going down a different road.
Thank you. And we have another question directed towards Dr. Gubert. Could you please elaborate on your gut-brain axis research?
yes, definitely. So as I mentioned, we have been targeting the gut microbiota to see if we can modulate the behavior, right? So we assume that we have a communication amongst the gut microbiota, the gut, the brain. So how do I target the gut microbiota? I use antibiotics, I use fecal matter transplant, as I mentioned, prebiotics, probiotics, and also environmental interventions. For example, exercise and diet can also modulate the gut microbiota. So all of these interventions, we are aiming to ameliorate the behavior and the outcome in specifically, as I mentioned, I'm working with Huntington's disease and schizophrenia. We are also interested in the relationship between the host and the gut microbiota, the population by itself, and also how these modulate the gut immunity. Because something that we are about to submit is unpublished yet, but we showed, we will show that the fecal matter transplant to Huntington's disease is not being very successful. And we hypothesize that's because we have a huge gut inflammation in these Huntington's mice. So what makes the engraftment of this new gut microbiota that we are transferring not being able to engraft. So that's very interesting, and that shows how important the host genetics, but also the host immunity, how this is important. So fecal matter transplant a few years ago was thought to be a very key intervention, but now other labs and also us, we are showing that is not, is more related to all the environment and how the host also handles this intervention. So yeah, we are, trying to use different techniques, but also trying to understand the host ability to modulate all these interventions.
I don't know, is it okay if I hop in and like add to that really quick? Just because innate immunology is like my favorite thing in immunology in general. But yeah, I mean, like I remember for one of my first classes, I wrote, it was a host microbe class, and I wrote about schizophrenia and the gut microbiome. And there, for example, for schizophrenia, there is a really high increase of Candida albicans in their blood or in their gut microbiome compared to the healthy average person. And what's interesting about Candida albicans is we all have it in our system. Most of the time it's commensal, but at times with certain triggers, it can become pathogenic. And so basically looking at that, like it's super interesting also because Candida albicans can elicit a certain immune response. So there are a couple of different kinds of immune responses. There is a Th1 mediated immune response, a Th17, and I know I'm throwing out a lot of jargon right now, but that's immunology kind of for you. But they're basically just different ways that our immune system will respond to different bacteria. So like, is the bacteria, or I guess in this case fungi, is it yeast? Is it bacteria with like a hard cell wall, or is it just depends on the type of bacteria, and it can skew either way. And so for schizophrenic patients, one of the hypotheses that I think people were throwing out there is like maybe Candida albicans is getting into the brain, and when it gets into the brain, then that causes this Th17 mediated response, which then can cause neuroinflammation and cause maybe more synaptic pruning than should be caused, and neurodegeneration in that sense. The other thing I was just going to mention too is that basically one thing that has been noted, and I don't think this is published yet, is that schizophrenic patients tend to smoke a lot of cigarettes, and they were finding that that's actually potentially nicotine is ameliorating that Th17 mediated hyperinflammation. And we're learning that from those patients, which we used to, maybe this gets back to the social part too, where we used to judge them for smoking cigarettes when really like they were giving us a super big clue as to what was going on. So I think that's also a really interesting thing to think about.
Yes, no, definitely. The gut immunity is so interesting because, we have only one wall of cells in the gut. So in an army of gut immunity that all the time constantly is trying to define, is this a good thing or is this a bad thing? Is this a bad thing we have to fight against to protect the host? So sometimes you have an imbalance in this definition, what is a good thing and is a bad thing. When you have this, you have, for example, inflammation, gut inflammation disorders or autoimmune disorders. So our gut immunity is all the time being challenged and can define, the way you respond to some diet compounds. So that will set if you are lactose intolerant or gluten intolerant, but also define, how you will respond to all of these microbes, including fungi, but also gut bacteria. And as Jackie was mentioning, even this balance and how you define what it is good and what it is bad in a few disorders are different, and then can be related to also how the phenotype of the disorders are. So yeah, very interesting.
Thank you. Jenna is currently trying to figure out what they want to do for their senior thesis for neuroscience and would like to research Alzheimer's in a lab setting, but don't know how to narrow down what or how they want to study it, and are asking if any of you could provide advice on how you narrow down what to research, or if there's any exciting new research they could work off of?
So Jenna, I think starting at Alzheimer's is a good thing because there's lots of research to be done in this field particularly. So as I touched on a little bit earlier, Alzheimer's disease in general is super, super complex, and the research that's been done and is emerging in the coming years is also very complex, and there's still lots of research to be done. So it's just kind of determining if you're interested in the causes, the preventative mechanisms, the pathology, new treatments, or maybe just understanding the efficacy of current treatments. So technically, I studied the theory of cognitive reserve, which essentially just says that individuals who have an enriched lifestyle have developed a large size cognitive reserve, which makes them less susceptible to age-related decline and the cognitive deficits that we do see in Alzheimer's. But this is still just a theory, so it needs some support. So do a quick Google search, see what's currently being studied, what's out there, what's supported, maybe what hasn't been supported recently, and try and find out why. So you can pretty much start in any corner of Alzheimer's disease that you want, and there's always something to be learned and always something to be added to.
Yeah. Oh, sorry, I didn't mean to answer that.
Thank you. Benjamin asks if in your respective geographic areas, has there been any research developments on the relationship between the immune system, COVID, and the brain that goes beyond identifying the blood vessel damage?
I can take this part. So there is a few labs that were explaining at least the loss of smell and how that was working in terms of COVID, because that was one of the symptoms that people could tell, is that their olfaction was not working, and olfaction is just a fancy word for smell, pretty much. This is more hypothesis driven, but what I'm particularly working on is very similar to kind of what COVID is potentially doing beyond the blood vessel damage. So my research is about basically the innate immune response to tau-mediated neurodegeneration and how it contributes to that. And so I think like one of the key papers, and I would highly recommend people checking him out, is Rudy Tanzi. So he studies, he came up with this theory called the antimicrobial peptide hypothesis of neurodegeneration in Alzheimer's. And that is basically about how, I think you guys might be familiar with herpes virus and how it gets into the brain, and how they found that that can cause neurodegeneration, or at least in cell culture, it can cause neurodegenerative phenotypes. Additionally, though, like what I'm particularly interested in is inside of these like amyloid beta plaques, which is what a lot of Alzheimer's patients, well, they have it. If you have Alzheimer's, you will have these plaques. But also, if you don't have Alzheimer's, you will have some plaques as you age. And what's interesting about it is there have been some studies that have been done where they found that those plaques actually, like sometimes in the center of them, are things like fungi, things like candida, things like herpes, things like just any other kind of trash that would be in the brain that's not supposed to be there. So one hypothesis is that what's really going on is maybe amyloid beta is an antimicrobial peptide, or at least it is functioning as both that and I think it regulates sleep in some ways too. But essentially, I think COVID, because we know it can pass through the blood-brain barrier, that's going to be something that we'll have to see as people age with this research, because that's the one downside to studying these kind of diseases, when you're having new viruses or like new things like this pop up, and we're asking questions about how does this affect lifespan? How does this affect like age-related diseases? And we're just not going to know until it happens, you know, until we start to see neurodegenerative phenotypes for people. But yeah, I think that that's pretty much that's at least for as far as I know, like what studies have been going on.
Right. If I could just jump in super quick, in relation to vulnerability to Alzheimer's disease from the pandemic and social isolation, I can kind of see a link possibly emerging, because if we think that positive early life experience or midlife or even late life experience is beneficial and protective against age-related disorders and Alzheimer's, then this can work in reverse too. So I think there lies a strong link in the social isolation and the stress that's come along with the pandemic and depression. And so there was actually a study that was published recently that showed a link between individuals who have chronic depression are much more likely to get Alzheimer's disease. And this is because of atrophy in a brain area called the hippocampus, which is important for learning and memory. And this is very vulnerable in depression, but it's also the first area of the brain that's affected by the Alzheimer's pathology. So there definitely will be some consequences, I predict, maybe coming up a little bit later in life, from the stress and the social isolation and the depressive symptoms, making your brain a little bit more vulnerable to later dementia and Alzheimer's disease.
Something that I am also interested in, and I have one PhD student working with, is the fact, the transgenerational effect of COVID infection, the paternal one. So we give pretty much the SARS-CoV-2 infection in males. And then after a few weeks, when they recover, we mate them. And then we analyze in F1 and F2 generation, the offsprings and also the grandchildren, to see the effect on behavior in these mice, and also how can they respond to immune activation, to see, you know, because we are thinking all the effects that we are having at the moment, but sometimes we are not thinking all the effects that we have in our future right? So in the offspring that we have, like next generation, how a COVID will affect that. So yeah, we have a lot of studies happening at the moment with COVID, of course, with maternal infection as well, but we are specifically working with the paternal effect.
And also, if I could just pop the word, because you guys are so, like, this is so interesting. I'm happy to like talk about it. I think also like the sort of chronic isolation and depression, we also know that there's hyperinflammation with depression. And there's also metabolic differences due to behavior. I mean, it's all looped back, right? Because you might eat less healthy if you're depressed. You might also feel more chronic stress, which to me is one of the biggest indicators of health, I think, at the end of the day, is how chronically stressed are you and how much chronic inflammation are you experiencing over your lifetime, right? Because it just builds up. And so I just think that there are a lot, like the paternal thing that you're doing is super cool. And I also think like linking that to maternal stuff as well, we know already that infections in mothers or certain skewed immune profiles in mothers can actually elicit, is actually correlated with like autism in children. So for example, we know that having a TSM.
And schizophrenia.
And schizophrenia, no, that is true. And I think I would wager, honestly, like most of diseases, because we're developing, and one of the key things, right, is like your mother has to be able to develop an immune system that will not attack the baby, and that shifts everything. And so if you have a secondary infection, you can sometimes end up having, you know, these sort of psychological disorders or other disorders that we were maybe not even aware of. And like one other thing I just like kind of wanted to point out as well, which I think is just that in general, like I think we're just going to see a lot of fallout from COVID in a lot of different in a lot of different immune ways, because the heart's affected, the lungs. I mean, I think everyone's heard about the what's it called, like long-termers or like long haulers. Why are those people like not able to recover? It's because their hearts are damaged, their lungs are damaged, and it is been so longly believed that the brain does not have an immune system and is isolated from that, but it's not. It'll just take longer to appear, likely.
Definitely. And also besides, and also together with the immune profile that can be modulated, the gut microbiome is very related to the immune. profile, and also social isolation. It was already shown that it is a model of depression in mice. For example, if you leave the mice for a few weeks isolated, this is a model of depression. So imagine for people how that is. And also it's related to increase in immune system, and also with change in the gut microbiota. So everything is related in a not a good way. So it can be a very negative loop that will amplify the phenotype and make it, even worse. So yeah, we will have impacts in our life that we are not even aware at the moment. We'll see in a few, years from now or in the next generation.
Thank you for that answer. How can a researcher in the field of neuroscience become financially independent of their research work?
Not sure if I understood the question. Can, I don't know, perhaps elaborate a bit more? Like,
I apologize. I can ask that the person who asked that question DM me or
okay.
elaborate, I suppose. Then another question would be if there are any particular books you would suggest for neuroscience and psychology.
So many. There's so many cool books. Kind of depends on the level that you're at. So one of the books that I really, really loved about maybe not neuroscience, but more of like the microbiome was Ed Yong's book, which was called I Contain Multitudes. I think that's like a really good introduction to some of this. Another book that I love is Time, Love, and Memory, which is a thing about Drosophila work and the history of that. And I think also there's a bunch of neuroscience books that are kind of like, it depends on like where you're at, like what you want to look at, like the social ethics of it. There's like a lot of neuroethical books, and I can post that in the chat. I can't remember it off the top of my head. And there are also a lot of books that cover more of the psychological end, which like The Man That Mistook His Wife for a Hat by Oliver Sacks. All of his books are really excellent if you're interested in like psychology. As for neurodegeneration, I'm trying to think about Alzheimer's books, but off the top of my head, I can't think of one specifically that I have looked at. So I should know that though. There should be more. So if anyone else has one, I would love to hear it.
Yeah, about the gut microbiota mod, that's a bit of my field. The John Cryan book, The Psychobiotic Revolution, is very interesting and explains a little bit how the gut microbiota can communicate with the brain. So if you're interested, that's a good way to start.
Yeah, all those books are great. The Brain That Changes Itself is a good one. That's more based on neuroplasticity and how you can kind of rewire your brain. So that one's a bit more psychology based, but that's a pretty interesting read, as well as all the ones that you guys mentioned.
Thank you. Here are some more questions for Dr. Guber. Are differences in gut bacteria populations genetically predetermined to some extent? And why do people with neurological diseases have different gut microbiomes?
So yeah, this is actually the million-dollar question. We are still in the infancy of the study of the gut microbiota relation with neurodegenerative disorders mainly. But the thing is, a few things differentiates the gut microbiota profile. One of these, it is definitely genetics. The other one is the immune profile, and also the environmental effect. So if you compare to twins, they will have a similar gut microbiota profile. But if they live in different houses or in different countries, that will be different as well, because the environment, the diet, if they take antibiotics. So the gut microbiota is very, very plastic and can be modulated in very, very many different ways. So yeah, the options are endless of how you can modulate and how that can be differentiated.
Yeah. And I also just wanted to like add to that too. There's like the genetic factor, right? Like you might have genes that would cause you to have like a lining on your epithelial or endothelial cells within your gut that would cause a leaky gut, that would cause hyperinflammation. There's also just lifestyle choices with certain psychiatric diseases that will definitely impair things. Like we've kind of covered some of it, but like exercise, huge. Like someone asked about stem cells before, they found that the cardio workouts actually help regenerate new neurons a little bit more than like other sorts of exercise. And so I think that it's just interesting to think about that. And then also interesting to think about, you know, what our society kind of forces people into doing, right? Like, so pills that people might take or medications might actually change the immune system. I mean, we know that for a lot of antipsychotics, right, they do change the metabolism in humans quite a bit in terms of weight gain and stuff like that. So I think that there's a lot to be looked at in terms of the microbiome and how that gets affected by those medications as well. And like, again, if you're somebody with Parkinson's, say, like you are not producing enough dopamine anymore, that means that your gut microbiome is just not going to have access to dopamine. And also serotonin, which is mostly produced in the gut, is going to be super dependent on your microbiome being healthy, and then also your own immune system being able to respond to that in a healthy manner.
Thank you. Once again, another question for Dr. Gubert. What are purinergic receptors, and what are the mechanisms by which these receptors relate to treatment for neurological diseases?
So purinergic receptors, they are based on the idea that ATP that we studied in our, even in high school, they were supposed to be inside of the cells, right? And they are the main energy source of the cells. But on the 70s, it was discovered that this ATP can also be in an extracellular environment. And then they can be even molecules of communication between cells. So we have receptors that can be activated by ATP. So those are purinergic receptors. The one I was studying particularly was the P2X7, which it is activated with a lot of ATP. It is in the extracellular environment, and it is usually associated with neuroinflammation, because when you have a lot of ATP in the extracellular space, usually that means that you have some cellular death happening, because we have a lot of ATP in intracellular space. So if one cell dies usually in a necrotic way, which means it will just explode and everything that it is inside will be in the extracellular space, that will activate P2X7 receptors in the neighborhood, and that will start signaling of neuroinflammation, and then can be related to some psychiatric disorders, for example, with bipolar disorder, which was what I was studying. So I had a pharmacological model of bipolar disorder, the mania-PO, which is giving amphetamine for 7 days for these mice. And then what I did was pharmacologically, I blockade this receptor. And then when I gave amphetamine to these blockade mice for this specific receptor, the hyperlocomotion that we expect that amphetamine will cause didn't happen, showing that this P2X7 receptor was very, very crucial for this behavior. So also I use knockout mice, which are mice transgenically who don't express this particular receptor. When I gave amphetamine to these mice, they also didn't show this manic-like behavior in these mice. So that was how I suggested that this specific receptor was supposed to be a very interesting therapeutic target for bipolar disorder. That was pretty much my PhD in 2 minutes explained.
Thank you. Some more questions for you. Do you use germ-free rat models for your research? And if so, how do you make them germ-free? And what ways do you assess the gut microbiome?
I'm loving all your interest about gut microbiota. It is definitely a very, very interesting topic, right? So I don't use germ-free mice for a few reasons. Mainly because germ-free mice, as you can imagine, they are already born without any germ in their body, not only in the gut, but also in their skin. They are literally germ-free. Firstly, you need a very special apparatus and a different way to handle these mice. So you need in your institute at least one floor specific for these germ-free mice, which we don't have in all institutes. So this is one of the reasons I don't have in the institute that I work with. But also, germ-free mice, they are related to, they have a lot of different phenotypes, because as you can imagine, the gut microbiota is related to lots of different things. So it's very hard to dissociate. So how for example, the immune system in these mice, because they don't have germ-free, how this is related to whatever scientific questions I'm working on. So I'm working with plasticity in the hippocampus, right? But how can I dissociate that this germ-free mice also has a different immune system? So we have very, very different factors that can affect and be limitations in this model. So what I do, I use antibiotics, for example, to deplete the gut microbiota, to specifically target, how a depletion of the gut, specifically microbiota, will interfere in the brain. So these mice still have the microbiota in their skin or in other tissues. So I think it's a little bit more clear model to study. Of course, germ-free mice are very important to understand other levels, and it was very important to show how important gut microbiota is. But yeah, we have to be aware of all the limitations of this model as well.
Well, and also just to hop in, because I'm about to be doing an experiment about making like germ-free Drosophila, actually, by giving them antibiotics. One of the ways that we can measure if they are truly depleted of the antibiotics or depleted of their bacteria is by squishing up the fly and plating it and seeing what grows, and hopefully nothing should grow on that plate. And something I just wanted to touch on too with the gut microbiome and the brain is that during development and during we were talking about with autism and viruses maybe affecting that in the maternal or paternal infections, we actually don't know. So the brain for a really long time was suspected to not have an immune system. It was supposed to be immunoprivileged, right? But now we're starting to realize there are a lot of molecules that actually double up as two different functions, right? So there's one gene called NMERI, and this modulates both sleep and similar to A-beta in that way, it modulates sleep and it has antimicrobial peptide, or it has antimicrobial function. So I think we have yet to understand, because we've delineated things so much into oh, this is an immune molecule or an immune cell, or this is a developmental molecule or a developmental cell, when in reality, we don't know that. That's just kind of how we've been studying them. And I think that the fields are sort of starting to cross over, where we're starting to recognize, hey, because the brain doesn't have an adaptive immune system, how does it deal with infection? And we're learning that, because the brain takes up so much space and energy, that a lot of these other sorts of proteins or molecules might be hijacked or evolved to do multiple different processes. And so I think that's really I think exactly what you said, germ-free established how important the microbes are. But if you even look at somebody who is not experienced many germs in their lifetime, that's more prone to, they get more prone to autoimmune disease. And so these are all things to consider when you're thinking about how to design your experiments and what conclusions you can draw from them. But yeah, sorry to banter on. I just think it's so fascinating.
No, please, thank you. And actually, just to complement we are always, when we study basic science, we are always trying to translate to humans, right? That's why we are modeling all these diseases in mice, for example. And germ-free mice is not easy to translate because we don't have people, unless those few people that have to live in bubbles, that don't have access to, but you know, if you have an antibiotic protocol, it's easier to be translated. And also the neurodevelopmental part is also important, because when you use germ-free mice, they never had microbes in their system. So this is also important for their development, right? So if you want to study in adulthood, whatever you are studying, I believe using antibiotic protocol is easier to understand this specific period of life. You are muted, Sydney.
Thank you so much. Anthony asks, do you use any deep learning or computational modeling to assist in your research? Because they're interested in computational neuroscience and neuroinformatics and would love to hear more if it's done.
I guess computational neuroscience is like a pretty big field. I can say that I've used some computational stuff for looking and analyzing fly behavior in the model that I use. So the model that I'm using is basically a humanized Drosophila fly with a familial. So this is like a hereditary tau mutation that causes very early neurodegeneration. So it's a primary tauopathy. And basically, I think that so for that, I am now assessing okay, what are their neurodegenerative phenotypes? And in order to characterize those phenotypes so that I can say that this is truly a model of human disease, because you have to make sure that, this mutation that's knocked into this fly actually does cause neurodegeneration. I've used some computational stuff to analyze their behavior. So one assay I've done is this locomotion assay called Flygram, which is fly group activity assay. So I just throw the flies in. There's 10 flies in each arena, and then you film it, and you can basically, through some computational methods, both just get an idea of how many flies are moving at any given period. So what I do is I put the flies in, give them just air, and then I give them an odor after a couple of minutes so I can see their response to an odor cue. And this is really important because olfaction is actually one of the first things to go. So sense of smell is one of the first things to go in a lot of neurodegenerative diseases. And so by doing that's also a computational method, because it's recording how many flies are responding at that given time. And then I can go back through the videos, and I've had some people do this with me. And you can look at things like behavioral phenotypes that are related to social aspects of, or social, I guess I would say, deficits in neurodegenerative disease. So being more anxious would be being around the sides of the arena a lot more. They're not as exploratory. They might chain a little bit more. One of the other labs I work in studies alcohol use disorder, and they do a lot of computation with that by seeing hey, if we get the flies drunk, what happens? And what's really interesting is they have a very similar profile to exactly what humans have when they drink. So they first get very hyperactive, then they sort of start to lose locomotion, and then they pass out. And so you can then look at things like inhibition of behavior. So males will try to mate with other males, and that's all done through computational work, because what Drosophila are really used for is screening for the most part. And because they're just such a short-lived animal that you can do that, and you can do it with high numbers, and that can give you more computational stuff. And with the microbiome, I mean, that's tons of computation.
Yeah, pretty much Jackie, she summarized all my life, which is pretty much analyzing videos by using computational softwares to analyze all the behavior that I do. So you can, with mice, you can analyze pretty much everything: anxiety, behavior, depression, also cognition impairment. And then you use these tools to be able to interpret and have a beautiful graph in the end. But yeah, also for analysis in the gut microbiota, we pretty much take the thesis, we send them to be sequenced, and then you have to use computational softwares as well to be able to analyze the 16S profile of these communities, and then be able to separate and see which taxa or genus, depends on the level you go. So yeah, a lot of computer work.
Thank you. In terms of, let me rephrase that question. Yeah, what are each of you most looking forward to exploring in your future research, both in the short term and long term?
So for me, I'm still at the beginning, so I have lots to look forward to. I firstly am just excited to see what comes out of the end of my master's thesis. So far, we're seeing some promising results, but like I said, we're a bit behind because COVID, we're getting small mouse numbers. So we're hoping once we top those numbers up to see more clear results and get a bigger picture on what my thesis is all about. So short term, I'm interested in that. I'm also interested, hopefully, when we can actually attend conferences in person. That would be very exciting for me to attend. A little bit longer term, seeing where the PhD takes me. I'm hoping to stay in the same lab, but maybe shift projects a little bit to see maybe if there's a way for me to incorporate what I've learned in my master's to some new information that I'll be learning in my PhD. So for me, it's a little bit shorter term, not so career focused, more so just educational based, but that's what I'm excited for, and I'm interested to hear what you guys have to say as well.
So yeah, for me, shorter term is definitely finishing all the current projects that I have, which will give me a better idea of how important the gut microbiota is for both Huntington's disease and schizophrenia. As soon as I have this answer, I will be able and better positioned to suggest a few potentially therapeutic targets that we can use for both those diseases, which are devastating and definitely need some new therapeutics. So long term for me, as I mentioned, I'm still an early career researcher. But I really enjoy and I really want to stay in academia. So what is the next step to me is to become more independent. So I'm still have a boss who funds all the experience that I am leading. So my next step would definitely getting a fellowship and a big grant to be able to be more independent, and long term potentially becoming a lab head and yeah, a professor at some point.
Yeah, so I guess for me, that's actually something I've been thinking about a lot lately. What I'm really excited for, like in the research realm, is that people are coming together that normally wouldn't have come together before to share like their science. So like I had never heard a neuroscientist ask me about immunology before basically this year, and now everybody is very interested in it. And I think that is just so exciting to see how these fields are coming together. And I will say it again, even though I might be pushing it a little bit, but it's an ecological perspective. And I think that that's starting to become a bigger thing. And it's also, it's a nicer sort of way of thinking about pathology, right? Because it's not just medication, it's not just lifestyle, it's not just microbiota, it's not just what you eat or who you see or what you do, it's all connected together. And in some ways, I think that's really beautiful. And there are ways to tweak things. For me personally, I have recently gotten really into sort of philosophy of science. So like I'm very interested in self versus non-self. And there's this book that came out by Thomas Perdue, who's basically a philosopher of science, where it talks about immunology and also the nervous system, and how both of these systems are determine self versus non-self. Like your brain is what determines that you are you and someone is someone else, but your immune system also does that. And so it's sort of bridging the connections between those things. And I'm hoping that I can get into some sort of thing where like I can think about those things, because I really love like thinking about ideas. I'm an idea-driven person, a big picture person. When it comes down to like nitty-gritty details and like lab like protocols, I'm just not as it's not as exciting for me. But you know, like I'm just really excited to keep pursuing this and keep seeing like where the research goes, and based on my past, I'll end up somewhere good just by being curious and invested in what I'm doing. So yeah, and also if I have like a piece of advice for anyone, it's just like keeping an open mind and like reaching out to people, because that's how I've found all of the things that I've been interested in. And they've kind of all collided into one, and I hope to keep that sort of collaborative aspect that I've developed because of just the broad range of things I'm trying to tie together intact. And yeah, I don't know what that looks like yet. So maybe I'm just excited to pave my own way a little bit.
Thank you so much. Here's a significantly more general question. Do neuroscience and neuropsychology careers vary from country to country? And if so, how do they vary?
So perhaps, yeah, because I came, you know, from the other side of the world, and now I am here, I think I can perhaps mention a little bit. So it doesn't change that much. I think what changed the most is how you'll be funded. So for example, in Brazil, the universities or the institutions, they pay for your salary. So you just have to apply for grants to pay for your research. In Australia, it's different. You have to apply also for your own salary every few years. And I believe in US is similar. So yeah, in Brazil is different from the rest of the world, I believe. No, actually Europe, a few countries also, institutions, they pay for your salary. So this I think is the main and is very important difference, because imagine every 4 years, even my PI, my boss, my professor, he has to apply for his own salary every 4 years. So a lot of people, they leave science because they can't be bothered, you know, or they need more stability, which sometimes, you know, in science you don't have, because you have to be always being outstanding and being doing good science and, you know, publishing a lot and applying for grants all the time. So it can be a bit exhausting. But again, you have to be passionate to really want to keep pursuing your goals and be applying for all of this. So see, I'm still here. I still want to be in science and academia, even having to apply for my salary every couple of years.
Yeah, I can only speak to the graduate student employment at this time. But in Canada, at least at my school, when you apply and you get accepted, in your acceptance letter, there's an outline of TA-ships and just a makeup of a stipend that you get for doing your research there. So you do get paid to do your research. You then apply for scholarships and grants every semester or so. And there's also a chance, depending on the project that you're working on. So the project that I'm working on, the grant that we had for it was quite large. So there was an opportunity for my supervisor to offer me some extra money from the grant to work on the project. So not only does it vary from country to country or school to school, it even varies within labs and within projects in that lab as well.
Yeah, and I would say like another thing to consider is I know that like in Germany, for example, in France, they have like you take your master's first and then you would go to your PhD. And I think it's free in Germany to get your master's. One thing I will say is with your PhD here, at least at Brown, and I think this holds true for other places too. If you apply for a PhD and you get in, it's usually fully funded. At least in my experience, that's been fully funded. At Brown guarantees you 5 years of funding. And that's just like a stipend that you know you're going to get. But in other places, but if you pass your qualifying exam and say you don't, that's usually like year 2, and say you're just like, I'm just not interested in research, you can actually just get your master's if you drop out, and then you wouldn't have had to pay for a master's. So little tips and tricks here. But anyway, I don't think you should do that like on purpose, but like it is an option for people. The other thing I was going to say is like cost of living is going to completely change what the salary is. So for me, I make the same amount of money as somebody who lives in San Francisco. My rent is just not nearly the same amount. And so that's, if you're going into like any sort of science career, just making sure that you know, like, if we're talking about money, at least, that you know like what the cost of living actually is there, because it cannot line up sometimes, and you're like, oh, this amount of money for here is great, but I can't even like afford to rent anything at this other place. So yeah, I think that's mostly it. And like, one benefit to being in the US is that it's just so big that you have so many people that you can collaborate with across a lot of places. And like in the Boston area, right? Like there's just so many schools that I collaborate with, and that's really nice. I don't know if that's really answering the question, but hopefully even regionally, it's just super different.
Thank you. And here is basically the last question. Would each of you mind sharing your biggest tips or words of advice for undergraduate students or even ambitious high school students regarding neurological and who are interested, given that they're interested in neurological and psychiatric disease research?
Well, I think the big tip that I would say, even being the perspective that I am now supervising students, is try to find an internship that you can, just be in the environment of a lab, to have the sense of how a lab is, be a shadow of a PhD student. And this gives you a lot of knowledge and a lot of, you learn a lot, and also give you the sense that it is really what you want to do. So yeah, I think that would be my main tip. Join a lab, offer your time to be, doesn't matter if it is 20 hours a week or 5 hours a week, just to be there and get a sense of if that's really what you want to do.
Yeah, that was exactly what I was going to say. But adding on, trying to have some private conversations with the lab members to see what the atmosphere is like in the lab. I've heard of some labs that there might be a little bit more competition. Some labs are a little bit more friendly and welcoming and would allow you to work on alternate projects with them. So kind of just talking to however many people you can talk to, getting your foot in the door in the lab, doing some research, seeing what you like, seeing what your strengths and your weaknesses are, and just kind of trying to make sure that the lab that you're in is actually a good fit. So for example, when I was in my third year, and I was thinking about grad school, I joined a human cognition lab, and I just found it not very exciting. I found it a little bit boring, but it was a learning experience, and I learned maybe this isn't quite what I'm interested in. So then the next semester I tried out working on mice, and that was the end of the story, and that's where I still am today. And the lab there was very welcoming, very friendly. There was never any seniority or competition, and it felt mostly like a family and like a team. So definitely trying out different labs, seeing the research that you're interested in, talking to people, talking to the PI, see if they have any space for you or what their upcoming projects are. And that would be my advice.
Yeah, so I mean, my advice for high schoolers, you are starting very early. That is very cool. I would say there are a couple of things that you can do. If you are a high schooler, you can look into, I know Brown has some programs where they have like high schoolers that come in and do some research with people on like certain special grant programs. So if there's something around that, like for you, then or in your area, I would definitely try to search those out. And I would also say be bold. So with PIs and professors, you can cold email them. You might not get a response, but you miss every shot you don't take, right? So I would say like there are a couple of things with that. So say you're really interested in the gut microbiome and the gut-brain axis, like I was so fascinated by it. You can find the person that seems, or papers, just by Googling it. And if you don't have access to those papers yet, because you're not, in a PhD program, or you don't go to a college that has access to those papers, one really good way is to just be like, hey, I saw this paper title. It's coming from your lab, and usually the last author on the paper is the one that you're going to want to email, because that's the PI and or corresponding author. And you can just sort of say like, can I have a copy of it? They can email you that if you want to read those papers. And then even more so, just saying like, hey, like I am interested in what you're working on because of this, and this. And you really have to be specific and think about it and make it personal. Otherwise, it's one of the worst feelings to get a generic email about somebody wanting to work in your lab, because you feel like, okay, this person doesn't really care. The second thing I would say is be persistent. If you really like that, email that professor maybe once every 2 weeks for like a couple months, or like maybe once a month for like a couple of months, and just keep saying like, I'm really interested. Do you have any other, and if they don't respond, you can even ask like, do you have any other like resources, or do you know of any other professors that are taking students or have resources where I could get more involved, because this is like my dream, or whatever it is. And honestly, I have seen that happen at least 4 to 5 times now, where people weren't going to take a student, but then they did, because they were like, this person has emailed me 5 times. They really want to do it. I believe in them. Like they could come start doing it. So with high school, it's going to be hard because you're not over 18 usually. And like lab rules are, it's tough because of liability. But I would say getting in touch with college professors, like that shows so much ambition. And if you can read those articles, anyone's welcome to email me. I think I put or tweet me or whatever, if you need like help like figuring out how to contact them. But like I think that that's a great way to just start your networking, and people will find that very, very like cool. People like talking about their research, and they like when people are interested in it. And the PIs that aren't liking it, and if you don't get a response, don't feel bad. They're mostly just super busy, and they don't even respond to my email. Like sometimes my own PI doesn't respond to me, and I have to be like, hello, like, so just remembering that, like, don't be afraid. Like most of them are super nice, and you just have to bother them a little bit and show like that you really tried to learn what they were studying.
Yeah, and also just to complement from a supervisor perspective, it's also very good for us to have a sense of how the student is before accepting them for like honors, masters, of PhD, because it is a commitment of years for a PhD, you know. So we also prefer to see if the student fits in our lab, if can survive the lab, if knows exactly how everything works, and if he's really interested. And receiving emails that Jackie said very well, that don't send a general email, because we always know, okay, this person is sending for 10 different people. I am just one of them. They usually won't respond. So you have to be very specific and personal, saying why you are interested in this lab and this line of research. But yeah, just saying that supervisors, they love having students, you know, for internships, because then they usually can say if they can accept later on for a PhD or for masters or something. So it's a win-win relationship.
Oh, sorry. Thank you. Many participants are wondering if, thank you, Ms. Howells, are wondering if you would be willing to supply your email, LinkedIn, or other contact information in the chat below.
Sure thing.
Yeah, and also if you guys the other thing I would say is on Twitter, like I know that Twitter's not always the best place to be, but that's how I actually started to figure out like the connections and network really hard on it. Follow find a scientist that you really like. Say you were into Dr. Gubert's research. Who is she following on Twitter? Who am I following on Twitter? Those are going to be indications of the people that are related to our research, and then you can read more about what they're doing. You can reach out to them on Twitter. You know, oftentimes professors are excited to respond to stuff on Twitter. If they're on there, that's kind of part of it. So yeah, I think that's looking at who your heroes are is a really good way to sort of get into that network.
Yeah, thank you. I acknowledge that we're going over time, so thank you so much for all of your time.
Yeah, thank you everyone for joining. Thank you to all our wonderful panelists for taking the time to help everybody out. But yeah, that's it. Thank you for attending this panel.
Thank you
Thanks
for having us.
inviting. Yeah, it was a pleasure. Bye.